anabenous symbiosis.
Well, I'd like to begin this morning by thanking the organizers for the invitation to come back to India again, as well as the opportunity to present this talk today. This is my 5th time back to India now, and with the generous hospitality of the Indian colleagues here, it's beginning to feel as though it's my second home. The system that I'll be talking about today is the Azolla-Anabenous symbiosis. And given that this symposium is on Cyanobacterial Biotechnology, I think it's very appropriate that we have a discussion about the Azolla-Anabenous symbiosis, given its wide range of agricultural and industrial potential. This particular system has the ability to serve as a sole nitrogen fertilizer source for rice crops. It's a completely organic biofertilizer, as most of you know. It can also be used as a fodder or food supplement, often replacing about 20% of the starchy food requirements for pigs, ducks, sheep, cattle, as well as being able to be used for culturing of freeling fish. It can serve as a weed suppressor, as well as reducing potentially mosquito populations. And it can also serve to improve water quality by removing nitrates, phosphorus, as well as heavy metals. So you can see that when we're thinking about biotechnology and types of applications, that this particular symbiosis has a great deal of potential. I would like to begin by just briefly giving an introduction to the different components of this system for any of those who are not familiar with it. Could I have the first slide, please? Shown here is an Azolla frond. The general arrangement of Azolla is such that it's free-floating and composed of multi-branched stems, and each of the stems contain pairs of deeply bilobed leaves. Next slide. This is a schematic drawing showing the location of the cyanobacteria. As I say, it has deeply bilobed leaves, lower ventral lobe, which is fairly thin, achlorophyllous, and it rests on the surface of the water, above which is the aerial dorsal leaf cavity, which contains the endosymbionts, which I should say consist of both bacteria as well as cyanobacteria. Next slide. This is a scanning electron micrograph showing the cyanobacterial cells within a leaf cavity. And in this association, each of the partners contributes. Azolla fixes and supplies the cyanobacteria with a carbon source in the form of sucrose, and in return the cyanobacteria carry out nitrogen fixation in the specialized heterocysts and fix enough nitrogen to satisfy both its own requirements as well as the requirements of Azolla. I should mention that the bacteria that are found in the association based on immunoelectron microscopy studies, certain types of those bacteria also contain nitrogenase and therefore have the potential to be contributing to the nitrogen fixation capacity of the system. This simply shows Azolla, as it's frequently used, being intercropped with rice. Next slide. When we look at this particular system, I think that it is an error to believe that it is all homogeneous. And the reason that I say this is if we look at the particular development of the system, we can take a main stem and going from the apex to the older, more basal leaves, if we look at the type, the cell type of cyanobacteria, as well as the bacteria that are present in these leaves of different ages, we see that there are each leaf is a micro-environment with very different things occurring in it. Could I have the next slide, please? The cyanobacteria more important than that. Indeed, the currents of the symbionts, the cyanobacteria, as well as the bactobionts, is an interesting and complex story, but one which we need to complete characterization of, with the ultimate goal of being able to, once the partners are identified, that for the purposes of agriculture and industrial applications, to create a zola that have greatest potential for those different needs. Thank you.
Here's Ricky Bauer's talk is open for discussion.
I have a simple question. Is it possible to reassociate the components back into symbiosis in vitro?
Yeah, the question is it possible to reintroduce the cyanobacteria back into the symbiosis in the laboratory? Is that correct? Yes. Okay. It has been extremely difficult to do that. There have been many attempts, and in fact, the only groups that have been successful have been in China and in the Philippines. And the key has been those reintroductions have only been successful if you, during the sporulation of the megaspores, the megasporocarps, remove the indusium cap, which contains some of the cyanobacteria, transfer it to one at a similar stage of development that is cyanobacteria-free, and then allow them to concurrently grow. And so there have been hybrids created in that way. But as far as other types of direct introduction of cyanobacteria through micromanipulations and so forth into the leaves, that has not been possible.
And the second question is, did you have background problem with your gold labeling?
Sorry?
Did you have background problem with the gold labeling?
Okay, the question is, were there background problems with the gold labeling? No. Clearly, in doing the labeling, you need to optimize the blocking and the protocol, but in doing so, I would say that you can get very high specificity.
Thank you. Dr. Ryan? Dr. Ryan?
Sandra, this in situ hybridization you did where you saw that heterosis had this visco labeling, right?
Yes.
These filaments, were they from the apical portion or the basal portion or the middle portion?
Would you repeat the question, please?
Okay, the question is, in the in situ hybridization experiments using as the probe the gene for carboxylase, whether or not the cyanobacterial filaments that were used in this experiment were taken from any particular region, apical versus basal portion. And the answer is we did not do that series for carboxylase. We did basically squashes, and so we did not have the profile for that, which would be of interest. The only thing we did the profile for was nitrogenase, and in which case even the heterocysts in the oldest leaf cavities contained.
Okay. Even so, though you took a squash, can you just say from which part you took the leaf on the front for this particular experiment?
No.
Because it would be interesting if it is from the apical region.
Yeah.
There could be some rationale, there may be still some probes maybe interacting because the gene may be there. But maybe from the middle portion or basal portion. Middle portion, I would say that should be changed.
What I can say, though, just to address that, is that in looking at many fields, in all instances, we saw the same labeling. So I would probably assume that if there were significant differences, or differences to the extent that in some instances the heterocysts did not contain transcript, that we would have seen that, and we didn't. But the exact levels, this isn't so quantitative.
With lights, please put the lights away so the direction you can see what's happening. There is one hand that's been up for a long time. If it's a quick question, go ahead.
Thank you, Mr. Chairman. If we go through literatures of 1960s 60s or 70s, then we most often see that the more is the heterosis frequency, the more is the nitrogenase activity. Now, in your slides, you have shown that though in the older NEPs you had higher heterosis frequency, you didn't get nitrogenase activity. Now, what do you think? What's the reason behind it?
Okay, I think everybody heard the question. I would say that, well, in the older leaf cavities, what we do know is that the heterocysts that are there have transcribed the NIF genes. The transcripts are there. But through the immunostudies, we know that nitrogenase is present. So we know that the protein is there. They've been translated. And so you have a situation with heterocysts that would appear to have gone through the trouble and effort to be able to fix nitrogen not fixing. Now, my answer would be that possibly there's a lack of a source of reductant or something that is not being supplied to the heterocysts in those old leaves that is inhibiting them from being able to carry out nitrogen fixation. So clearly in Azolla, the answer is not, let's find out how we can make more heterocysts, but instead I think the question is how can we ensure that the heterocysts that are there, or how can we activate the ones that are already more than halfway there?
Okay. Well, all right.
Excuse me, we have to go on to the next talk. We're really over time.
Okay, I'll discuss with her later on.
Okay,
Thank you.
thank you.
So the next talk is by Marcus Geisler, Willie Conan, Jutta Richtner, and Jürgen Schumann, with Marcus Geisler giving the talk on expression, purification, and properties of the Senecicystis 6883 calcium ATPase, functional differences to eukaryotic calcium-dependent ATPases.
Thank you very much.
I'll ring the bell at 18 minutes and then a few times at 20.
Okay. First of all, I would like to thank the organizing committee for the opportunity to present our results on the Senecicystis calcium ATPase. In the beginning, I would like to present some data on some properties of the enzyme produced from sequence analysis. Then I would like to show an efficient expression system of the Senecicystis calcium ATPase in E. coli. Further, some data for biochemical characterization of the enzyme, and special emphasis will relate on the functional differences of this prokaryotic enzyme to eukaryotic calcium ATPases. Next slide, please. In the beginning, I would like to show some pictures from our town, Düsseldorf, where all these experiments are made. Düsseldorf is situated in the west of Germany at the River Rhine. This is part of downtown of Düsseldorf. Next slide, please. Like this university, our university is called after a German famous poet. His name is Heinrich Heine. You can see him on the picture here. Next slide, please. This is our botanical garden, and over there, this is our institute here. Next slide, please. Okay, this is the object I'm talking about in the self-made bioreactor. This is Senecicystis PCC6803, and this is a strain for which the complete sequence information is now available. We got yesterday for several times. And if we put some hydroxylamine to that phosphoenzyme formation product, we get no signal. This is a direct proof for this kind of chemical linkage. Next one, please. Of course, we did phosphoenzyme formation with the expressed ATPase, which is much easier. They are separated not in the Lambda type of page genes, but they are separated on async. But we see a strong, we also introduced you to the in vivo overexpression mutant, and this shows from the growing just a really different phenotype, but this is still brand new, so I cannot say too much about that.
Further questions? Yes.
Marcus, could you explain the inhibition action of hydroxylamine on the calcium ATPase activity?
Yeah, it was not, I explained, the question was if I could explain the hydroxylamine inhibition of the ATPase activity.
Why?
Okay, we did only, and this is the, it's not a real inhibition. I mean, the kind of linkage between the aspartyl phosphate and the labeled phosphate is an acyl phosphate linkage. And the hydroxylamine is just a chemical reaction using, I think it's called an alkalic reaction, just to, it's not a real inhibitor, just a chemical one.
Yeah.
Correlation.
We found, you mean protein kinase?
Yes.
We found typical, say, AMP dependent protein kinase reaction site, and we tried to using eukaryotic type of thrombin combined to find a radioactive labeling, but we couldn't identify. So normally these enzymes contain several sites for PGA or PGC, but nobody knows about these prokaryotic enzymes. We couldn't find something from our experimental data.
Another question. You mentioned that this protein probably is involved in signal transduction. Is there any event known where this particular enzyme is necessary for?
This particular?
Yes.
In this organism, you know, they are not that valuable. This is new, and there are only some sequences found in different strains.
Not physiological.
And maybe there are described 5 prokaryotic calcium ATPase enzymes. So no one knows. And the major problem is that still it is not sure whether calcium could work as second messenger in prokaryotic cells. But for me, it's quite clear because the use of calmodulin in cyanobacterial strains gives an indication for that.
How many ATPase do you have in the system?
How many genes? Only one. There's just one.
And how many different types of proteins?
Didn't get it. How many different types of?
Of ATPase as proteins do you have? You know?
No, I didn't get it.
In
How many?
6803, how many different types of ATPases do you find according to the sequence?
Only one.
No.
There must be non-calcium ATPases.
Besides,
Besides the calcium ATPase, do you have some other ATPases?
No, there's just one calcium ATPase gene in synthesis. In Alabama there.
as a protein,
The amount of protein
sodium, potassium, ATPase, maybe.
of this P-type. There's,
Yes.
yeah, of this P-type enzyme, there's only one.
Maybe we can discuss it during the tea break further. Any more quick questions? If not, thank you very much for a very interesting presentation. The next presentation is going to be by the chairman of the session, Professor Peter Vogt. We have known Professor Vogt for so many exciting discoveries in almost all spheres of cyanobacterial biology, especially for the development of some very elegant, novel molecular genetic techniques by his laboratory. Today is going to tell us all about heterosis development and what may probably be the shortest talk he ever gave. Professor Vogt.
That's used in many places in the world. I've seen very few papers from India that use it, and so perhaps a reintroduction would be helpful. From there, I'm going to go on to discuss in broad terms what we know about the process of differentiation of heterocysts, and I'll finish up by asking for some help and hope that someone may be able to help. Okay, may I have the first slide? It's there, and this shows the entities that are involved in transfer of DNA. Is this microphone working? No. Can we have this microphone working, please?
Yeah, it's working.
It's working. Okay, good. It's all right. Now it's working. Okay, good. Thank you. So imagine that this is an E. coli cell And down here you have a little bit of a corner of Anabina showing. And the system for transferring DNA involves a conjugate plasmid such as RP4 or RK2. And I'm repeating some of what our Dr. Reddy said yesterday. So the function of this plasmid is to encode all the functions that are necessary for two bacterial strains to mate together. It is normally able to transfer itself. Such a plasmid can normally transfer itself to a second bacterial strain. In the case of Anabina, in fact, as far as I know, really, in the case of any cyanobacteria, assuming that it does transfer itself, it does not then replicate in the new strain. There's what here is called a shuttle vector, but more generally is a cargo plasmid. A plasmid which is what you really want to transfer to the recipient Anabina, or whatever other cyanobacteria you happen to be using. And it's such a plasmid might be able to replicate in the cyanobacterium, or it might be that it would have a piece of cyanobacterial DNA and should recombine at its homologous place in the chromosome of the recipient. It might bear a transposon, in which case it would not replicate in the cyanobacterium, but the transposon might transpose. For some plasmids, the conjugate plasmid can simply transfer another plasmid. That process is called mobilization. That doesn't always work, and for PBr322 derivatives, it does not work. And so, for at least some PBr322 derivatives, one needs another plasmid that adds a function which really is probably just that it nicks one strand of the DNA, so that strand is single-stranded, and the single-stranded DNA then moves into the recipient strain. And finally, and quite important, and this has developed in recent years, we use methylase plasmids, the point being that there are restriction endonucleases in Anabina, in many cyanobacteria, whose function is to recognize DNA as foreign and to initiate the destruction of the foreign DNA. But if one methylates the DNA at the particular sites that are recognized by the Anabina restriction enzymes, then that DNA gets in without being harmed, without being cut. Let me have the next slide, please. I'll skip some of the other people at this point. Thank you for your attention.
So that 20-hour program has been completed in 20 minutes. So all this paper is open for discussion. Questions? Yes, please.
Well, anyway, I got a chance to ask him why certain cell of Anabina differentiates into a heterocyst. This is a question that we usually ask, scientists usually ask, but to my belief, it is probably the cell division pattern in the Anabina, and this is my assumption based on the fact that in gliotrichia, what we have seen is that the basal cell of the hormogonium, which is released, that is the youngest cell of the group, and that youngest cell differentiates into a heterocyst. So, is it possible that along the hormogonium of the Anabina, there are some cells due to division pattern, after certain number of cells, the youngest cell occurs, and that cell differentiates into heterocyst?
I think this is an excellent question. The question, if I can rephrase it, is if you start with a filament, all of the cells of which appear similar, how is it determined upon nitrogen step down which one or which ones will differentiate? It's very clear that once certain cells start to differentiate, that they will inhibit their neighbors from differentiating. But nonetheless, the question is which ones at the very first start to differentiate. And it seems that the hypothesis that the questioner was raising is an excellent possibility, namely that it has something to do with the cell division cycle. That, to my opinion, has not been proven, but it seems to be an excellent hypothesis. There's a little bit of there is a bit of data from Wilcox's laboratory that showed that it was in a particular Anabina continuum, the smaller of two daughter cells of a division would be the one that subsequently would differentiate upon nitrogen step down. But more generally, indeed, it may be something to do with the cell division cycle, but what that would be has not been clarified.
Polysaccharide.
Aerobic nitrogen fixation. From some theoretical studies by Tony Walsby and also experimental studies, it seems clear that the major barrier to the entry of oxygen into heterocysts is the glycolipid layer that lies inside of the polysaccharide layer. That glycolipid layer, however, is probably quite fragile, easily broken, easily damaged. And so it seems to be that in the strains in which the polysaccharide layer is affected, the glycolipid layer, as a consequence, gets damaged, and as a further consequence, more oxygen can get in, and then one has nitrogenase inactivated, so one loses aerobic nitrogen fixation. Is there any idea how cells are sensing the need for nitrogen? Rama Subramanian, do you think of any answer?
Dr. Rama, you had a question. Would you like to react also to this? Yes, sir.
We've also looked at the what happens upon nitrogen step down. We put reporter transposons randomly in the chromosome, and we look for genes that are activated rapidly upon nitrogen step down.
Is involved in sensing the nitrogen to carbon ratio in the cell and would be involved in heterocyte differentiation too. Has been found in Synechococcus and also in Pterophrix, which is a strain which differentiates heterocytes, and I think that it could be also true for Anabina.
I take this opportunity to thank Dr. Wall for a very interesting otherwise very pleasant conference. But I hope the little bit of stress they say induces some creativity, and I look forward to that. What we do is study stress responses of nitrogen-fixing cyanobacteria, mostly Anabina strains. And 6-7 years ago, when we began this kind of study, there were very few labs in the world doing such studies. Today, the number has expanded, and we also find, even within this meeting, that all sorts of different approaches are being used, which is good because it will generate a lot of basic information. What we do is study stress proteins, and it may not be the best approach to know what we are looking for, but it has certainly given us some interesting information, and I would like to share that with you. The two Anabina strains that we use mainly are shown in the first two slides. This is a freshwater strain, native to the paddy fields in the institute where I work, Baba Atomic Research Center. We call it Anabina L31. This is a salt-sensitive but a highly osmotolerant strain. The next slide, please. This is what we call Anabina turulosa. It's a brackish water strain, again isolated from the coastal areas within our institute. We call it Anabina turulosa. This is probably what Peter is looking for, mechanics next to heterosis. I don't know if it has other features that you are looking for. The next slide, please. We take these wonderful microbes and subject them to all sorts of stresses. I don't want to go into what we call stress, but the stresses that I'm going to talk about today, I'm sure all of you would agree, are stresses. And what I want to share with you are our results on salinity, osmotic stresses, a little bit on heat shock, and if possible in the end, something to do with potassium deficiency, which we feel may be a somewhat global stress for cyanobacteria. The next slide, please. Well, just to show that many stresses, of course, result in gross structural differentiation, formation of heterosis or acyanides that you have seen, some of which can be visualized by fluorescence microscopy like this, which shows lack of phycocyanin-based chlorophyll fluorescence in heterosis and presence of it in vegetative cells. But what we are looking for is more subtle changes, what we think are molecular differentiation, shown in the next slide. And next slide, please. And this would describe the approach that we use. What we do for analysis of these stress proteins is subject the organism to different length of stress or different concentration or level of the stress. And in the last few minutes, usually 1 to 5 minutes of the stress period, we label it in vivo with 35S methionine, extract the proteins, run it on gradient gels, dry the gels, take an autoradiograph, and look for the proteins which picked up the label during those 5 minutes, meaning the proteins which were either synthesized during that period or the proteins whose synthesis was repressed during that period. This is a slide that shows you the salinity stress-induced proteins in one of the Anabina strains. One thing you can see here is that the controls themselves undergo changes with time because they are differentiating or because they are doing different things at different times. So at each time point you have to have an unstressed control present to know what is specifically induced by the stress you are applying. The response is sometimes very, very rapid. Within 5 minutes you can see certain proteins disappearing, many new proteins coming up. In case of salinity and osmotic stresses, such protein changes are somewhat persistent for an entire period of 6 hours here, or in many experiments for 5 days. The synthesis of such salinity stress or osmotic stress-induced proteins continues throughout the stress period. The next slide shows you that sometimes these changes are somewhat very, very short-lived or transient. That's the few slides which we believe suggest that potassium has a very important role to play in stress tolerance in these organisms. Starting with salt or osmotic stress, we have now seen it happening in almost all stresses. Potassium itself is very important for growth, nitrogen fixation, photosynthesis, everything in this organism. Over here is a potassium supplemented or a deficient strain which lacks the epiprotein of nitrogenase. Within hours of adding potassium, the epiprotein comes back, nitrogenase activity comes back, and revival of the culture takes place. The next slide. We have seen the potassium deficiency induce proteins in these organisms, and we find with days, the severeness of stress increases. On day 2, you have synthesis of several so-called potassium deficiency proteins or PDPs in this strain. The number is about 15. I would like to add that in E. coli, where only 3 such proteins have been seen so far, namely the KDP system, there are no other such proteins known either in bacteria or plants to our knowledge. We may be ignorant about it, of course. In cyanobacteria, we have seen many, many of them. The next slide shows you, we can skip this, let's go to the next slide. These are the expression, just last 2 slides. The expression of many of these is very, very need-based. You have them induced under potassium deficiency. Within 10 minutes of adding potassium, most of them disappear. The next slide. The next slide. The next one. And the last one. Yeah, I will close with this. This was a model proposed by Mizuno for expression of KDP system in Anabina, the 2-component as well as for heat shock signal, all of which of course was dehydration and changes in membrane stretch, and that seems to be responsible for expression of KDP. The only thing I would like to change is KDP is probably but only the tip of the iceberg. We have at least in Anabina 15 other PDPs to look at, and we can just change this KDP to PDP and the model will still hold. I would like to close here, and thank you very much for your attention.
It's time for questions. Yes, you do.
If you investigate stress situation, you have to deal with population, and you also have to concentrate that there's a cell cycle. Are there some experiments on the one side with synchronous cultures looking on the cell cycle and the response of the cell cycle to the stress? And the other question is, are there continuous cultures look on the steady states at the beginning and at the end? Because in this, if you use batch cultures, you always in a transition state in a different way.
Answer to both the questions is that we have ourselves not studied either of the possibilities that we are talking about. We have not studied anything in continuous culture, but we have not seen any cell cycle dependent or cell cycle dependent expression of these stress proteins. We have done it at several stages of growth, several stages of division cycle, and we have not found any particular difference. But in turbidostats or in chemostats, we have not looked at.
Okay.
you have isolated feedstock protein. Do you find homology with the eukaryotic feedstock protein?
Yes, the reason I called them grow EL or HSP70 is because we have detected them using antibodies obtained from anti-HSPs from ECOVAN.
How transient are they?
How transient
is
are
the effect?
the heat shock?
Yes.
Heat shock is the most transient of stresses that we know of. You see them within 30 seconds, some of them, and you see them, most of them disappear in about 30
So
seconds.
when you couple it with sucrose, you have to do it within that time.
Yes, I didn't have time to go through all those things. If you pre-treat with heat and then you subject it to sucrose, what happens is that, but for those 4 common proteins, other heat shock proteins disappear instantaneously. You have synthesis of 12 other osmotic stress proteins of OSPs. What we find is if you are pre-treated with heat, it enhances the level of OSPs in a subsequent exposure to sucrose. Now, get through many slides. We have again used the antibody for KDPB obtained from Professor Eltendorf. I skipped some of the westerns if you saw, and that does pick up this signal very strong. A 78 kilodalton protein, it picks up in anabina under all sorts of potassium deficiency conditions.
Okay, but because we have also looked, but we can...
One small thing I may add, that we have used both the KDPB and KDP-ABC antibodies of Professor Eltendorf, and we pick up with both only KDPB and no KDP-A or KDP-C proteins in the roster.
I'm afraid we're running over time, so we're going to stop questions now. Money, you have some comments?
I'd like to thank all the speakers as well as the chairperson and co-chairperson. Although Professor Peter Oak would continue to chair the next session on biofertilizers, he'll be joined by Dr. B.D. Kaushik as the co-chairperson. We would like to present a small memento to Professor Peter Oak and to Dr. Apte for chairing the sessions. Dr. Sundari will give it to Peter Oak, and Dr. Malliga will give it to Apte. So we adjourn for tea and reassemble in 15 minutes time.
And I come from the University of Bharathidasan in Sri Lanka. The rice field ecosystem, wetland rice fields, offer a very congenial environment for the growth of algae, what we would call a bloom of algae. In this case, it was a mixture of mostly Aphanothecae and Pleotrichia. The next slide we have, guys, this way. These are the things that are found attached to the buds and the surface soil. The next slide. Here you see epiphytic algae, epiphytic on weeds. Next slide. Epiphytic on rice plants. Now, these are of course specifically blue-green algae. Many of them are nitrogen fixing, but on the other hand, if you look at the rice field situation, the rice field ecosystem of course harbors a lot of other algae. It's a complex situation of green algae, diatoms, blue-greens, both nitrogen fixing and non-fixing, and we must understand this situation properly if we have to manipulate it successfully. And therefore we took an initial step of looking at a natural succession of algae, all the types of algae, greens, diatoms, blue-greens, in a rice field without any interventions. And the next series of slides will show this kind of thing. Next slide please, immediately after land preparation. Possibly due to the what you call the priming effect of liberation of nutrients from the virgin soil that has been left fallow, as well as the addition of fertilizer. As the rice canopy grows and the rice seed start putting out their pillars, with the effect of shading, as the effect of shading, most of the green algae tend to diminish. And they are substituted generally by the non-heterocystous, unicellular and filamentous blue-greens. The next slide please. You see the Cyclorias which are undifferentiated, right? Spirulina Again, sometimes you see them together with and if no fertilizers, if no nitrogen fertilizers are added, then you get series of heterosexual species, Cylindrospermum, next slide. Gliotrichia, mostly epiphytic, next one. These are two older, next one. Chitonema with false branching, next one. Astroplatus with two branching, both systems in series, next one. And even Tiruchirappalli, the multifaceted in every rice field. This is a collection of soil samples we have observed and taken photographs to show that the variety. But the sequence is the heterosister species. Now this is if you allow this to happen without any interventions. And that kind of succession I think should be understood very clearly if you were to successfully manipulate the system. Without such an understanding, if you just go and add inoculum, very likely it may not succeed. Then take them to the laboratory, grow them in nitrogen-free media or enrichment media as we call them. Then you get a different picture because you are selective and depends on what time you are collecting the soil. So this kind of discrepancy should be borne in mind when you compare results from different authors, from different publications, because many of these authors do not give a clear expression of when they collected the soil, what was the condition of the field at the time of collection. We conducted the, it doesn't matter. Well, towards we are almost full of achinates in a dry field. Next one. Similar achinates of caloties. Next one. These are achinates of Gliotrichia.